Hello, 9th standard. Have a good day. Today is our 10th video class that we are going to continue plant physiology. Heading photosynthesis. I hope you have heard, you have learned a little or you have learned a lot for photosynthesis. Okay. Now what is photosynthesis? It is said photo means light, synthesis means to build up. What is to be built up? That only we are going to learn. Shariya? Now you should know the equation 6 CO2 6 CO2 plus 6 water will give light. Here chlorophyll and here I write C6H12O6 plus 6. As this equation we will study one by one. Okay. Now photosynthesis, photo light again I say, and synthesis the light to be built up. As the light energy is converted into chemical energy by the process photosynthesis. Okay. For the photosynthesis to occur, we need 4 things. As one is light. First one light. Second one is chlorophyll. Second one chlorophyll. Third one water. And fourth one is carbon dioxide. I repeat, for photosynthesis to occur, we need 4 things as light, chlorophyll, water and carbon dioxide. Light and chlorophyll, they are essentials of photosynthesis. Idhilana photosynthesis nadakadhu. So light and chlorophyll, they are essentials of photosynthesis, and water and carbon dioxide, they are raw materials. Light and chlorophyll, they are known as essentials of photosynthesis, and water and carbon dioxide, they are the raw materials. சரியா? Now this is a green plant. Then this is a green plant. இது வந்து leaf உனக்கு தெரியும். Then this is also leaf. This is a green plant. Okay. In that water is absorbed by the root system. Carbon dioxide from the atmosphere, it is entering. Sunlight fall on it and chlorophyll present in the leaf. I repeat, sunlight falls on the leaf, chlorophyll present in the leaf, carbon dioxide from the atmosphere getting in, water is absorbed by the root system. So water and carbon dioxide, raw material, sunlight and chlorophyll are the essentials of photosynthesis. இது வந்து நீ நல்லா நீட்டா டயக்ராமேட்டிக்கா போடணும். ஒரு क्वेश्चन இருக்கு நமக்கு. Give the schematic representation of photosynthesis. As you will draw a potted plant. You will draw a potted plant and then water வந்து root system ல இருந்து மேல போடணும். Then இங்க carbon dioxide எழுதி இங்க arrow mark உள்ள போடணும். இது வந்து sunlight. Sunlight பொதுவா ரெட்டா இருக்குது. இது வந்து sunlight. அது வந்து leafல விழுது. This is schematic representation of photosynthesis. Okay. Now listen. This essential which will when the sunlight falls on the leaf, it is said it becomes excited. Excited, na? It will start reacting. So when the sunlight falls on the leaf, it becomes excited. The leaf will become excited, which is acting upon the chlorophyll. Now in the leaf, the chlorophyll is present in the structure called chloroplast. Chlorophyll is present in the structure called chloroplast. In the chloroplast, the sunlight viழும்போது it becomes excited, and during that time the water is reacting on it, where it is getting split into hydrogen and oxygen. So now the water is split into hydrogen and oxygen. Now this splitting of water in presence of photosynthesis is called photolysis. The splitting of water in presence of light is called photolysis. So now this hydrogen, as it is unstable and highly reactive, it has to be accepted by the hydrogen acceptor, that is NADP. That is NADP. It accepts hydrogen to form NADPH2. I repeat, first, during the process of photosynthesis, sunlight falls on the leaf. Leaf contains chlorophyll in chloroplast. The chloroplast becomes excited and acting upon water, which is absorbed by the root system, and that absorption is by diffusion, imbibition and osmosis. When it reaches the leaf, it is utilized for the photosynthesis, and that water undergoes a process called photolysis, which is splitting of water into hydrogen and oxygen. As this hydrogen is highly reactive, unstable, it has to be accepted by hydrogen acceptor, and that hydrogen acceptor is NADP, that is nicotinamide adenine dinucleotide phosphate. Nicotinamide adenine adenosine nicotinamide adenosine dinucleotide phosphate, and it is reduced to taking hydrogen in reduced form. Now this oxygen is used as a byproduct. Adola oxygen vela mudinjadu. So here in the process, first splitting of water, and then that is photolysis, then NADP is reduced to NADPH2. Two events, first it is taking place in the leaf. Now as it is taking place in presence of sunlight, these two reactions are called light reaction. In photosynthesis, these two reactions are called light reactions. Then further, things needed for photosynthesis, light எடுத்துண்டாச்சு, chlorophyll எடுத்துண்டாச்சு, water எடுத்துண்டாச்சு. Carbon dioxide எங்க போச்சு? After this formation of NADPH2, the carbon dioxide is taken by the plant, and then by further series of reaction, it is reduced to carbohydrate. And as it is taking place throughout the day and night, in whether the presence or absence of light, it is called dark reaction. So there are two reactions. One is light reaction taking place in presence of light. Another one, dark reaction. Throughout the day and night, reaction is taking place, where carbon dioxide is reduced to glucose, and that glucose on further synthesis, it is forming the starch. Okay. So the equation for photosynthesis, C6CO2 plus 6 water in presence of light and chlorophyll it forms C6H12O6 another glucose. And 6 what? Oxygen. This oxygen is evolved as a byproduct. I hope you are following. So the mechanism of respiration, it is a light reaction and dark reaction. இது வந்து நம்ம புக்ல இல்லை, ஆனா நான் வந்து கொஞ்சம் டீடைல்டா போறதுனால உனக்கு சொல்லி கொடுத்திருக்கேன் and you will understand. Okay, இதுக்கு வந்து क्वेश्चन ஆன்சரும் தரேன். இப்போ photosynthesis, where light is essential for photosynthesis அப்படினா there is an experiment where you take a potted plant you take a potted plant with a leaf, and in one leaf you cover a portion of the leaf with the black paper. You cover portion of the leaf with the black paper. So here, now leave the potted plant in the dark room for 4 to 5 hours and then keep it in the sunlight for photosynthesis to occur. What can you see? After 4 or 5 hours, you remove the leaf which is covered with the black paper and then test the leaf for stars. How will you test? First, the leaf, the black paper should be removed. Keep the leaf in boiling warm water for a few seconds and then in alcohol for few seconds and then you add iodine solution. If the portion where starch is prepared, it will turn blue black and there is no light available which is covered with the black paper will show no effect, showing that the portion which is exposed to sunlight will show that light is essential for photosynthesis. This is one experiment. Okay. Another one, you take a beaker where oxygen is evolved during photosynthesis. You take a beaker and then you keep a funnel. And over that, you keep test tube filled with water. And inside the funnel, we find cut branch of hydrilla plant. You will keep a cut branch of hydrilla plant. Okay. So now here, you take a beaker with the water, and then you take a funnel, invert it, and over that you keep a test tube filled with the water, and then inside the funnel you keep cut branches of hydrilla plant. Keep the apparatus in sunlight for some time. We find oxygen bubbles will be collected in the test tube. After 4 or 5 hours, you carefully remove the test tube and test the gas for oxygen by introducing a glowing splinter. It will burn brightly, showing that the gas evolved is oxygen during photosynthesis. Okay, so we are doing 2 experiments. One is light is essential for photosynthesis. That is called starch iodine test using the light and leaf and the black paper. Another one, this is called test tube funnel experiment. This is known as test tube funnel experiment where oxygen is evolved during green plant. Evolved during photosynthesis from the green plants. I hope you are able to follow this. So in photosynthesis, there are நம்ம புக்ல வந்து ரொம்ப டீடைலா கொடுக்கல, only 4 essentials தான். நான் கொஞ்சம் டீடைலா போய்க்கேன் to explain light reaction and dark reaction. I hope you are able to follow. Shall we continue? Okay. So this is for photosynthesis. முதல்ல ஒரு நிமிஷம் எக்ஸ்பெரிமெண்ட் பாரு. Test tube and funnel experiment and the equation. Now we will continue as photosynthesis. Photo means light, synthesis means to build up. So what is to be built up? As light energy is converted into chemical energy. Now where this photosynthesis is taking place? In the green leaf. The green plants which are capable of doing photosynthesis, they are called autotrophs. Green plants which are capable of doing preparing their own food, they are called autotrophs. Otherwise, the plants which cannot prepare their own food are non-green plants, or they are depending on other living or non-living organisms. They are called heterotrophic plants. And nutrition of plants which can prepare their own food, it is known as autotrophic nutrition. One process, that is photosynthesis. Whereas heterotrophic nutrition, there are different types as parasites, saprophytes, insectivorous plants, and symbiotic association. That we are not going to deal now. So now this photosynthesis, what is the equation? Says 6CO2 plus 6H2O in presence of light and chlorophyll. C6H12O6 plus 6O2, where oxygen is evolved as a byproduct. The end products are glucose and oxygen. This glucose, on further synthesis, it is forming starch or carbohydrate. Light and chlorophyll, they are forming essentials of photosynthesis, whereas carbon dioxide and water, they are as raw materials for photosynthesis. நாலுக்கும் difference தெரியும் நமக்கு. சரியா? Then you should know the schematic representation. The reaction that is taking place where light energy is converted into chemical energy, it takes place in two steps. One is light reaction, other one is dark reaction. And during light reaction, photolysis of water is taking place, where it is split into hydrogen and oxygen, and then this hydrogen, as it is unstable and highly reactive, it has to be accepted by hydrogen acceptor, that is NADPH2, that is forming NADPH2, nicotinamide adenosine dinucleotide phosphate. I hope you are following this. சரியா? The experiments என்ன பார்க்கிறோம்? One is light reaction. Light is essential for photosynthesis. Another one is test tube and funnel experiment, where oxygen is evolved during photosynthesis. There is one more, as we say, proton plant, foliage plant. Foliage plant, proton plant. Where will they do photosynthesis or not? Proton plant வந்து variegated leaf னு சொல்லுவோம். அதுல வந்து இந்த லீஃப் வந்து green color வந்து ரொம்ப டாமினன்ட்டா இருக்காது. மற்ற கலர்ஸ்ல அப்படியே கொச்ச கொச்ச கொச்சனா மல்டி கலர்ல இருக்கும். So they are called variegated leaf. அதுல photosynthesis நடக்குமா? Yes, it will occur. ஏன்னா every leaf will have chlorophyll dominant, whereas in variegated leaf, example coleus, proton plant, the chlorophyll is hidden, so it is capable of doing the process photosynthesis. Okay, so here in coleus variegated leaf, green plants can prepare their own food. So we should know the term autotroph. And then already சொல்லியாச்சு, like this essential and test tube and funnel experiment. I hope you are following. The next one, what you are going to do, இது வந்து photosynthesis. I hope you are able to follow. Shall I continue this? Just to check panel. Now here, today we are going to do biology worksheet number 5, scoring key answer. Already உனக்கு வந்திருக்கும் நினைக்கிறேன், இருந்தாலும் படிச்சிருக்கோம். Now question 1, photosynthesis அது அப்புறமா கண்டினியூ பண்ணுவேன். ஏன்னா ஒரேடியே ஃபுல்லாக உனக்கு தெரிஞ்சுன்னா படிக்கிறது கஷ்டம். இதுவும் ஒரேடியே படிச்சுட்டே இருந்தா போர் அடிக்கணும். சரியா? So 1A, meristose, which means divisible or having cell dividing activity. Then B question, parenchyma tissues are present in aquatic plants. parenchyma tissues are present in aquatic plants. Okay. Now name the storage material in potato and apple. And luminol. In potato, parenchyma vacuoles are filled with starch. In apple, parenchyma stores sugar. Name the storage material in potato and apple. In potato, parenchyma vacuoles are filled with starch. In apple, parenchyma stores sugar. Then example for tissues and organs in plants and animals. That is the first paragraph. In plants, the roots, stem and leaves are organs, whereas xylem and phloem are tissues. Xylem and phloem are complex permanent tissues. Similarly, in animal, stomach, for example, is an organ that consists of tissues made up of epithelial cells, gland cells and muscle cells. Then binomial of hemp and jute வந்து cannabis sativa, that is hemp, and Corchorus capsularis jute. Cannabis sativa hemp, Corchorus capsularis jute. Then differentiate diagrammatically scleroids and fibers. The diagram வந்து நான் வீடியோல வரும், நீ வந்து கொஞ்சம் பாஸ் பண்ணி போட்டுக்கோ. உனக்கு வந்து இந்த கீழேயே கொடுத்துருக்கேன் இந்த டயக்ராம். தெரியுதுன்னு நினைக்கிறேன். தெரியுதா? Right. Then this is for worksheet number 5. Then worksheet 6 வந்து give one word. Water containing tissue, xylem. Then this type of tissues are present in the center of the vascular bundle. Another pith. This type of tissues are present in the center of the vascular bundle. Another pith. Then tracheids will have lignified walls. Companion cells will have phloem. Tracheids companion cells will have phloem. And companion vandirukku adu maathikku phloem, right? Then differentiate between wood fiber and bast fiber. Last correction, bast correction panikku. Bast fiber. Xylem fibers, also called wood fibers, are elongated, lignified and pointed at both the ends. I repeat, xylem fibers are also called wood fibers, elongated, lignified and pointed at both the ends. Phloem fibers are bast fibers. They are sclerenchymatous cells, lignified and provide mechanical support to the plants. Then xylem thickenings are annular, spiral, scalariform and picted. Then label diagram, xylem element I already given either particle. Xylem. Clear theriyudha? So this is for worksheet 5 and 6. I hope you are able to follow. சரியா? So I already சொல்லியாச்சு photosynthesis. For 2 minutes sum up பண்ணிக்கோ. Then worksheet 5 and 6 நம்ம discuss பண்ணிக்கோ. Right? We will continue in the next class. Thank you. Next to photosynthesis, we are going to learn transpiration. What is transpiration? Just now I said it is the evaporation of excess water from the green leaves. What is this excess water? We are again I say when we water the plant, not all the amount of water, what you say volume or amount of energy, not all the amount of water is utilized for photosynthesis by the green leaves. Only water which is necessary for photosynthesis will be taken by the green leaves, and other excess water is evaporated in the form of water vapor. That is called transpiration. Now this transpiration, it is mainly taking place by a structure called stomata. In the stomata. Stomata, green color. It is present on the green leaf. Another green color. I hope green color clear bold. Now when we take a cross section of a leaf, we get structure like this. This is the upper epidermis. This is lower epidermis. center வந்து leafல இந்த bulge portion வந்து midrib region. சரியா? Now here we find a tissue called mesophyll. நான் அதை டீடைலா போல இது மட்டும் தான் சொல்ல போறேன். This is called mesophyll. Now this upper and lower epidermis is arranged like this. Now this structure is called stomata. Same way இங்கேயும் இருக்கும். இந்த மாதிரி arrangement இருக்கும். சரியா? So now this is the stomata. Now this stomata will have a kidney shaped cell called guard cell. Kidney shaped cell called guard cell. And surrounding that is the epidermal cell. நான் இது stomata சொல்றேன். So இந்த epidermal cell இருக்குல அதுதான் இது. So these are called epidermal cell. Now this stomata, the guard cell, they are having chloroplast and the nucleus. Now this chloroplast with the guard cell, kidney shaped structure with the nucleus and chloroplast. Now this center portion, by opening and closing of this guard cell, the exchange of gases is taking place. Evaporation of water is taking place. So stomata is doing two major functions. One is evaporation of water through the guard cells and exchange of gases for respiration and photosynthesis. Another சொல்லணும், for photosynthesis carbon dioxide is taken in, oxygen given out. For respiration, oxygen is taken in, carbon dioxide is given out. That is done by the opening and closing of stomata. சரியா? Now here the guard cell in the line வந்து thinner in nature, and the outer layer it is thinner in nature and inner to that is thicker in nature. And then the opening and closing நடக்கனால அதனால it is having a thicker layer. So when the guard cells are open, we represent diagrammatically like this. Whereas when the guard cells are closed, இந்த மாதிரி close up போடணும். This is closed, this is open stomata. சரியா? Now this transpiration, it is of three types. One is stomatal transpiration, second one is cuticular transpiration, and third one is lenticular transpiration. This is lenticular transpiration. Stomatal transpiration is through the stomata. Structure of stomata என்ன சொல்லிருக்கேன்? It is present on the leaf as upper and lower epidermis. More stomata will be seen on the lower epidermis than the upper epidermis. What is the structure of stoma? Stoma singular, stomata plural. Stoma it is having a pair of bean-shaped cells called guard cells. It has nucleus and cytoplasm. And surrounding the guard cell, it is called epidermal cell or subsidiary cells. Epidermal or subsidiary cells. Epidermal or subsidiary cells. By opening and closing of stomata, it is said for opening of stomata, more potassium ions from the guard cells are utilized. For the opening, the shrinkage of the guard cells. Guard cells will become shrunken, thereby opening the guard cells. And then when it is, otherwise we say, when it becomes turgid, when the guard cells become turgid, by more potassium ion, the opening வந்து பெருசாயிடும். And when it is becoming shrunken, then it is closed. சரியா? bulge portion வந்து turgid condition, shrunken வந்து flaccid condition. Two terms we come across. One is turgid, bulging, and shrunken வந்து flaccid. So that is how opening and closing of stomata taking place. I repeat, during stomatal transpiration, potassium ions present in the guard cells, they play a major role. When the accumulation of potassium ions are more, the guard cells become bulging, that is turgid condition. That time stoma becomes open. So exchange of gases or evaporation of water possible. And then when it is become flaccid or shrunken, then the stoma remains closed. That is for stomatal transpiration. Then cuticular transpiration in all xerophytes, that is open shear cactus plants, in all xerophytes, and then in most of the leaves, the upper epidermis is covered with the thick cuticular layer, cutin, deposition of cutin, that is cuticular layer. So transpiration is possible through the cuticle also. So that is cuticular transpiration. The next one is lenticular transpiration, where in the bark of woody trunks, old stems and shrub மாதிரி நிறைய bark சொல்லுவோம்ல, அது வந்து irregular இருக்கும். That will have openings. It is said in the bark of the woody stems there are opening or holes called lenticels. Through them also evaporation is possible. I repeat, through the bark of the or opening on the woody stem, the lenticels are present. Through that transpiration is taking place. That is called lenticular transpiration. So there are three types of transpiration. One is stomatal, second one cuticular, third one lenticular. As the name says, through stomata, through cuticle, and through lenticels. So yeah. Now this transpiration, when we say you take a potted plant, this is a plant, then you cover it with the plastic bag. Or plastic bag cover. You take a potted plant, cover the shoot system with the plastic bag and make a hole on the plastic cover. After few hours, say 1 or 2 hours, keep it in the sunlight for some time. After 2-3 hours, you see there are more water droplets on the plastic bag. Again, that is through the evaporation of leaves. This shows that plants respire through the leaf. Another example, that is called bell jar experiment. That is known as bell jar experiment, where it is a shape of a bell. You take 2 bell jars and take 2 potted plant. This is A and this is B. Take 2 potted plant. In one plant, you keep the leaf, shoot system with the leaf. In another plant, without leaf. And keep both the bell jar on the sunlight for some time. What can I observe? We find more water droplets on the bell jar and few water droplets on the bell jar. Idhula vandu leaf irukku. So water droplets ore possibility vandu leaf la irundha vandirukkanam. Seriya? So here more water droplets were potted plant with the leaves, whereas here without leaves la less water irukum, transpiration irukum, but it will be very less in nature. Seriya? So this is bell jar experiment for transpiration. Okay, now just go through this stomatal transpiration, cuticular transpiration, lenticular transpiration. These are the types of transpiration. Okay, now this transpiration, it is considered as a necessary evil. Evilனா transpiration நடக்கணும், அதே சமயம் அது வந்து ஒரு ப்ளஸ் பாயிண்ட சொல்ல முடியாது. ஓகே, ஏன்னா it creates a pull in leaf and stem. அந்த வாட்டர் மேலே போறதுனால it creates a pull in leaf and stem. And then it also creates absorption force. நிறைய வாட்டர் மேலே போறதுனால it has to suck more water. So it creates absorption force in roots. Then it is necessary for continuous supply of minerals. Transpiration is a must for a continuous supply of minerals. And also it regulates the temperature of the plant. It is also regulating the temperature of the plant. I hope you are following. So these 4 points are, it regulates the temperature, it creates force to suck more water, and then it is helpful in bringing the water up. So these are called necessary. Transpiration is a necessary evil. சரியா? So this is for transpiration. ரொம்ப சின்னது, நம்ம 9th book ஒன்னாக்கினதுனால the photosynthesis, transpiration எல்லாத்தையும் கொஞ்சமாக்கிட்டாங்க. ஸோ நானும் அதே என்ன புக்கில் இருக்கோ அதே சொல்லி கொடுத்துருக்கேன். ஆனால் photosynthesis நான் இன்னும் கொஞ்சம் டீட்டெயில்டாக போகிறேன். photosynthesis எக்ஸ்பிளைன் பண்ணோம்னே உனக்கு வந்து ஒரு explanatory part வரும். அதையும் you have to learn. Please understand. Are you clear? We will continue in the next class. Thank you. We will continue with the plant physiology, photosynthesis and respiration with some explanation of the figures. So far I have been explaining and describing the process of photosynthesis and transpiration. Now we will learn some portion with the figures. Now what you are looking at this figure is a schematic representation of photosynthesis. நீ என்ன லேபிள் பண்ணுவ? ஃபஸ்ட்டு பாட்டமில் கீழே ரூட் சிஸ்டம், அண்ட் தென் ஷூட் சிஸ்டம், அண்ட் தென் லீஃப், சன்னு, இல்லையா? இதில் ரூட் சிஸ்டம் இட் டேக்ஸ் வாட்டர் and then carbon dioxide is entering in. அதை வந்து நீ நான் லேபிள் பண்ணல, நீ தான் லேபிள் கற்றுக்கணும். And then sunlight falls on the green leaf, chlorophyll, and then இன்னொரு black arrow இருக்கு பாரு, அது வந்து oxygen is evolved. ஒன்று வந்து carbon dioxide is entering in, இன்னொன்று வந்து oxygen is evolved. That is schematic representation. And again sunlight and chlorophyll, they are the essentials of photosynthesis, whereas water and carbon dioxide are the raw materials for photosynthesis. These 4 are must for photosynthesis to occur. Edella sunlight and chlorophyll as essential, water and carbon dioxide as raw materials. Okay, then equation for photosynthesis, chemical equation for photosynthesis. அப்படியே தூக்கத்தில் கேட்டால் கூட உனக்கு சொல்ல தெரியணும். 6CO2 plus 6H2O in presence of sunlight and chlorophyll, C6H12O6, that is glucose, plus oxygen, 6 oxygen. This glucose on further synthesis is made into starch and carbohydrate. Otherwise we say carbohydrates and starch. And oxygen is evolved as a byproduct. This is chemical equation for photosynthesis. And as I explained yesterday, this photosynthesis, plant physiology, previous photosynthesis patti, adula the process of photosynthesis, it occurs in 2 phases. One is light reaction other one is dark reaction. The reaction involving chlorophyll, solar energy, and sunlight, water that produces ATP. ATP na expansion adenosine triphosphate. It is a energy rich compound. And NADPH2, that is nicotinamide adenine dinucleotide phosphate in reduced form. That is called light reaction. The light reaction, there are two events taking place. One is photolysis of water, where splitting of water into oxygen and hydrogen. As this H2 is unstable and highly reactive, it has to be accepted by hydrogen acceptor, that is nicotinamide adenine dinucleotide phosphate, NADPH2. அது வந்து சொல்லுவோம். Okay, and these two events, one வந்து photolysis, another in the reduction and ATP formation. ATP is a energy currency expansion adenosine triphosphate. This is happening during light reaction. In dark reaction, carbon dioxide, which is obtained from the atmosphere, is reduced to carbohydrate with the help of ATP and NADPH2 formed during light reaction. And for this reaction, light is not required. Throughout the day and night, this reaction is taking place. So light is not required for this reaction. Now what is observing is experiment to show that light is essential for photosynthesis. There are two leaf actually the green color blue. There are two, you are taking two green leaf A and B. One you are covering with the black strip, and another one after the test we are going to observe. Another, you keep a potted plant with the covering the leaf with the black strip. After some time, you keep it in the sunlight for 4-5 hours. After some time, you remove the leaf from the plant and then test the leaf for starch. I can take out the leaf and keep it in a warm water, and then in alcohol. Then you add iodine solution. The portion which is covered will remain as it is with no change, and the portion which is not covered will turn blue black. So B leaf la paru rendu blue black irukla adula exposed to sunlight. Another one வந்து yellow னு இருக்கு, actually அது green color ஆ தான் இருக்கணும். சரியா? This is light is essential for photosynthesis. இந்த black stripக்கு பதில் இன்னொரு பக்கத்துல ஒரு எக்ஸ்பெரிமெண்ட் இருக்கு பாரு. அதுல take a potted plant, அது ஒரு சின்ன ஸ்டாண்ட் எடுத்துக்கோ. அதில் வந்து there is one apparatus called light screen apparatus. அதில் வந்து இந்த லீஃப்க்கு நடுவில் ஒரு ஸ்டார் இருக்கும். அந்த apparatusல the star வந்து க்ளோசிங் த லீஃப், அது வந்து இந்த லீஃபில் கிளிப் பண்ணிட்டேன்னா அந்த போர்ஷனுக்கு வந்து சன்லைட் கிடைக்காது. ஸோ திஸ் இஸ் கால்ட் கேனாங்ஸ் லைட் ஸ்க்ரீன் எக்ஸ்பெரிமெண்ட். இந்த எக்ஸ்பெரிமெண்ட் apparatus கண்டுபிடிச்ச பேர் கேனாங்ஸ். ஸோ இது பேர் கேனாங்ஸ் லைட் ஸ்க்ரீன் எக்ஸ்பெரிமெண்ட். After keeping this potted plant in the sunlight for 4 or 5 hours, then remove the leaf and test for stars by adding iodine. They accept the star portion which is covered, other portion will turn blue black because the star portion is not exposed to sunlight. This given experiment is called test tube and funnel experiment to show that oxygen is evolved during photosynthesis. If you take a beaker with the water, then you take a funnel, over it you invert a test tube with the water. Inside the funnel you take cut branches of hydrilla shoots. Then after keep the apparatus in the sunlight for 4-5 hours. Now you can see bubbles are coming from the cut branches of hydrilla shoots, thereby releasing the gas on the test tube. Now carefully take out the test tube and test the gas for oxygen by introducing glowing splinter. Then it will burn brightly, showing that the gas evolved is oxygen. So this is called test tube and funnel experiment. Sorry, those two experiments for photosynthesis. Now this is transpiration, where evaporation of excess water through the leaves and the leaf may through the stomata. இங்கே வந்து ரெண்டு ஸ்டொமேட்டா டயக்ராம் இருப்பார். ஒன்று வந்து ஸ்டொமேட்டா இன் ஓப்பன் கண்டிஷன், அனதர் ஒன் ஸ்டொமேட்டா இன் தி க்ளோசிங் பொசிஷன், க்ளோஸ்டு ஸ்டொமேட்டா இல்லையா. அதில் the guard cell, yellow colors are called guard cell with the nucleus and cytoplasm. Surrounding the guard cells are the green leaf structure that is called epidermal cell or subsidiary cells. Now the stomatal pore open வந்து நல்ல broad-ஆ இருக்கும் பாரு. என்ன சொல்லியிருக்கேன், guard cells வந்து outer layer will be thinner and the inner layer will be thicker. Appo it is kept opened. And then when the more potassium ions are entering into guard cells, it becomes turgid, nalla bulging, and it makes the stomach open. Then when the gaseous exchange or evaporation of water is over, then the guard cells will become flaccid, shrunken, thereby closing the stomata. So this is called mechanism of stomatal transpiration. நான் வந்து டயக்ராமேட்டிக்காக கேட்பேன். explain the mechanism of stomatal transpiration diagrammatically அப்படின்னா உனக்கு இந்த ரெண்டு டயக்ராமும் தெரிஞ்சிருக்கணும். சரியா? இது வந்து பெல் ஜார் எக்ஸ்பெரிமெண்ட் ஃபார் demonstrating transpiration. What is it? Take two bell jar A and B. In one bell jar you take a potted plant and another one potted plant without leaves. So the apparatus வந்து keep it in the sunlight. After some time we find more water droplets on the potted plant A with the leaf, whereas less water droplets in the bell jar B. Leaf it transpires more water, whereas B bell jar where bell jar without leaf வந்து லீஃப்ல இல்லைனால ஸ்டெம்லேருந்து ரொம்ப மினிமம் குவான்டிட்டியில் டிரான்ஸ்பிரேஷன் நடக்கும். சரியா? ஐ ஹோப் யூ ஆர் ஏபிள் டு ஃபாலோ திஸ் எக்ஸ்பெரிமெண்ட், பெல் ஜார் எக்ஸ்பெரிமெண்ட். சரியா? இந்த வீடியோவை இன்னும் 2-3 தடவை போட்டு பாரு. ஏன்னா நம்ம புக்கில் இந்த டயக்ராம்லாம் இல்லை. ஆனால் நான் வந்து கிரியேட்டிவ் கொஷனுக்கு கேட்பேன். அப்போ வந்து சொல்லாதே மறுபடியும் ஐ வாண்ட் திஸ் அண்ட் தட்னு நீயே காப்பி பண்ணிக்கணும். ஐ ஹோப் யூ அண்டர்ஸ்டாண்ட். ரைட்? வில் கண்டினியூ இன் த நெக்ஸ்ட் கிளாஸ். ரைட்? தேங்க்யூ.